Well, it's a pleasure to be here. Always love to come to this side of the continent, I guess, because of Vancouver can't say the country, but one of the drawbacks of living on the West Coast is the fact that when I do go to the East Coast, that I always have to leave at like 5:30 a.m. or something like that. But it's all good. So thank you for your kind invitation. So today I'll introduce you a little bit to my world, biomaterials in the urinary tract. The title pretty much says it all. It's still an ongoing battle, and hopefully by the end of this talk you'll understand why. And so the idea is to sort of walk you through it. Excuse me. What's the problem? What have we tried? Why has some of it failed? Where are we going now? And then the idea is to sort of be able to show you what it's all about, so that when we have discussions after about the engineering side of things, which I will disclose right away, I have no idea about, how we can work together in the future. So to begin with, a couple of disclosures about being a paid consultant. Lead or co-investigator on some industry collaborative research grants through companies. But none of what I'm talking about today has anything to do with promoting products or anything like that. I'm just talking about the pure science. And the other thing too is in terms of biomaterials in the urinary tract, most of my talk will be on ureteral stents. And the reason that is that's kind of where I've done most of my work. However, everything that I talk about in the context of stents is readily translatable to Foley catheters because they all live in the same environment, which is the urinary tract. So in terms of, let's start with the definition of a stent. Well, there's two, actually, depending on what type of stent you're talking about. It's a device used to maintain a bodily cavity or orifice or contour after skin grafting. That's one way. An example of that would be, for instance, a vascular stent as well. And then the other is more like a ureteral stent, which is a catheter, rod, or tube within a tubular structure to maintain luminal patency or protect an anastomosis or graft. Now, I realize that is a lot of medical talk, but basically all it means is it's a tube in a tube, essentially. Okay. So this is basically what a ureteral stent looks like when it's placed. So we turn down that.
Maybe the font so many. Oh yeah, that's much better.
There you go. So the idea is, we call them double J stents. The reason we call them double J stents is because they have a curl at one end and a curl at the other end. The idea behind the curls is just basically that it gets anchored into the bladder and the kidney. So as you can see from the picture on the left here, unlike a vascular stent, which gets placed and then gets deployed and expands and pushes on the walls of the artery or vein, the ureteral stent just basically sits in the lumen of the ureter. For reasons that are yet unknown, the ureter says, okay, something's in me, I'm going to dilate. And that's basically what we want to do. So imagine if there is an obstruction somewhere along the ureter, placing a stent bypasses that obstruction and creates a little bit of an opening to allow urine to pass. Because if it doesn't pass, you get back pressure on the kidney, which will start expanding, and you get something called hydronephrosis. And of course, the kidney, just like many other things, can only expand for so long until it starts getting damaged. So the idea of a stent is to maintain patency, bypass an obstruction, and allow urine to flow somewhat freely.
And how big is this stent? What is the diameter and the size?
It's only a couple of, total diameter is only about 2 or 3 millimeters.
Okay.
Yeah, they're not very wide. They do have a hole in the middle. Now, the majority of urine flow is actually on the outside, so there's always discussion on whether or not the hole is actually necessary, but it's there for simplicity's sake. So what is the ideal stent? Well, the ideal stent has to be biocompatible, just like any medical implant, if you will. Has to be radiopaque, meaning you have to be able to see it on an X-ray. It has to relieve intra and extraluminal obstruction, so it has to be rigid, because if it collapses, it kind of defeats the purpose. And it has to be able to resist encrustation infection and cause little discomfort. Well, it's points 4 and 5 that are the big problem. And the reason I'm here today is because really there is no stent currently that encompasses all of these factors. The reality of it is that no biomaterial used in the urinary tract is able to completely withstand the effects of the urinary environment. It's so complex. There are many different factors, many different components found in urine. It's basically anything that our body doesn't want, or need for that matter. And so it makes it very complicated to try and put anything in it. So device-related UTIs, urinary tract infections, and incrustation are a significant cause of morbidity to patients. Mostly, obviously, because of pain and discomfort, but also because the only real way to treat, well, chronic UTI or even chronic incrustation is to replace the device, well, which means it's an additional surgical procedure depending on the extent of either the infection or the incrustation. Invasiveness is dependent on that as well. So these are major limiting factors for especially long-term use of biomaterials within the urinary tracts. So I'll walk you through the problems. And on today's talk, I'm only going to focus on incrustation infection. Rather than symptoms, that's for a different talk. So let's start with incrustation. And it's basically been generally accepted that the longer you leave a stent, or even a catheter for that matter, in the urinary tract, the greater the likelihood that it will encrust. And so this is a study published in the 90s that basically shows exactly that. Stents that were indwelling, this was looking at 299 patients, okay, and stents removed from these patients. So stents that were indwelling for less than 6 weeks, there's about a 9% of them were encrusted. And then that increases as the dwelling time increases. So by the time that a stent is indwelling for longer than 24 weeks, 90+% of them have encrustation. Now, the degree of it will depend, obviously, on various factors. Some, well, the majority of which are basically patient-related, and mostly the chemistry of the urine, of course. But why is this a problem? Well, there's two problems. One is if the incrustation is big enough, removal is going to be very difficult. That's one issue, but think about it too. The other issue is you have a rod essentially within a tube. Once the incrustation gets big enough, the tube, the ureter, gets obstructed. So once again, you have an issue. So that needs to be broken up and taken out. So again, we're talking about an extensive surgical procedure. Now, what are these incrustations? There's two different types. Okay, one is calcium-based, and these are the most common type of calcium kidney stone. It's a calcium oxalate stone. Okay, about 85% of kidney stones are calcium oxalate stones. And I suppose I should have mentioned that the majority, or most of the time, why stents are placed is because of kidney stone surgeries. Okay, and so calcium stones form mostly due to the urinary environment being supersaturated for calcium and oxalate. So when calcium and oxalate bind, they form an insoluble crystal. If oxalate binds with anything else, sodium oxalate for instance, that dissolves readily. But when it combines with calcium, it becomes a problem. Okay. The second type of incrustation is called a magnesium ammonium phosphate, and we call it STULite in the medical industry. And that is a type of incrustation that forms only in the presence of bacterial infection with urease-positive bacterial species. Now, why is that? So essentially, let's go through that step for a minute. And here in this particular scenario, we're talking about encrusting a Foley catheter. Okay, so bacteria start attaching to the surface of the catheter, they form biofilm. Now these bacteria produce urease. And so basically what they do is they hydrolyze urea, of which there's lots in urine, which then produces ammonia. Well, that increases the pH of the urine from normal, which is around 5.5 to 6, up to as high as 8 or 9. Now that results in the precipitation of calcium and magnesium, which then combines with ammonium to form these magnesium ammonium phosphate stones. And calcium malachite is another product. And so you can see quite nicely in the pictures here that over time, this is after 1 hour, you can see bacteria here quite nice, and then as time increases, you can start certainly to see crystals of screw-like forming. And the same here in the full picture at the bottom. Why are there two different sets of pictures? Well, these are actually silver and nitrofurazone-coated catheters. Okay, so these catheters do have antimicrobial surface coatings, but clearly you can see that despite all of that, you still have bacterial adhesion and significant incrustation.
So this is a result of like a healthy patient, or a patient is suffering from a specific. I mean,
well, they would have had a catheter inserted for whatever reason, and then they developed a urinary tract infection with a urease positive bacteria.
Okay.
So the only reason that incrustation happened is because of the urinary tract infection.
Okay, so you
Not
have
because
that
of
already
anything else.
to start. Okay.
Yeah. So now why struvite stones or incrustations are so difficult to treat is because the bacteria actually live within the incrustation itself. So you can take a stone, let's say, or an incrustation, break it apart, and you can see the bacteria in there quite happily. They're still alive. Okay, which makes it so important to have to remove every single fragment from the urinary tract, because if you don't, it can quite readily form another nidus for another episode. And these can quite easily, when urease positive bacteria infect the kidneys and cause pyelonephritis, you can get stones quite quickly that basically encase the entire collecting system, as we call it, so the entire inside of the kidney. So these are quite serious and significant. So there we are. So those are struvite incrustations. And so here,
So many days for us.
and of course, as a biomaterial, as a biofilm, the bad thing about biofilms is that bacteria don't just stay in one place; they just keep spreading, right? And so as more bacteria spread, they form another biofilm a little bit further up. This process just keeps repeating itself. And so here, again, you have pictures of several different types of catheters, some of which, again, have antimicrobial coatings, others don't, but you can see quite readily that here's a 12-hour picture for the catheters here, and then that goes over time to blocking the lumen of the catheter. So once again, we have a whole different problem because now the catheter doesn't function anymore, and the urine isn't draining properly anymore. So what kind of methods have been tried to try and prevent incrustation? Well, there have been several. People have tried to change the bulk material. For instance, silicone, which is has a low surface energy, and the idea was to because of that it's more resistant to incrustations. Or it's very surface coatings, different hydrogels, antibiotics, as we just saw, don't work. Okay? Pentazine polysulfate, and even oxalate degrading enzymes. The idea there is for the oxalate degrading enzymes to break down the oxalate to prevent the calcium oxalate crystal deposition. Another one that's been tried is coating stents using heparin. The reason for that was that heparin is highly negatively charged, and again, the idea was because of that significant negative charge, the incrustation wouldn't occur. In this particular study, you can see coated versus uncoated ureteral stent removed from the patient. And this study did show that there is a significant reduction in incrustation. Now that's only calcium oxalate-based encrustation, because we performed a subsequent study when I kind of entered into this field. My first study was basically taking different types of stents and testing the bacterial adhesion to the surface of these stents. And I can tell you that bacteria attach to the surface of this stent just as readily as they did to an acrylic stent. So as far as struvite encrustation would be concerned, this stent would be able to prevent that. So there we are. So let's look at infection. What's the scope of the problem? Well, here is a table that summarizes several studies that basically looked at stents removed from patients, and these stents were indwelling for various amounts of time. And they basically looked whether these stents were colonized with bacteria. Now, as you'll see, for the majority of these studies, except maybe for 1 or 2, these patients received antibiotic prophylaxis. So the idea was for the antibiotic prophylaxis to prevent bacteria from attaching to the stent. When you look at the actual numbers of colonized stents, there we go. The range is anywhere from 30% all the way up to 90%. And trimethoprim isn't exactly an easy antibiotic either. Okay, so clearly it's a problem. But one thing to keep in mind is that colonization, or the presence of bacteria, doesn't necessarily mean infection. Okay? Because infection, whether or not the presence of bacteria causes an infection, depends on, obviously, indwelling time, the level of colonization, but the most important factor is the immune status of the patient. Now, having said that, you could argue that, well, there's always a certain level of compromised immune system, because the patient would be sick in some way to receive the stent to begin with. So, there's always some sort of level. But still, it's the immune status of the patient that really determines whether the presence of bacteria actually causes an infection. Because the other thing, too, is the indwelling time. So even if bacteria are present on the surface of the stent, if they're only in there for 2 to 3 days, you could look at that as almost just a surface contamination, because you remove that stent before the bugs actually get to the point where they would cause an infection. Okay, so that's always something to keep in mind. Just because there are bacteria there doesn't necessarily mean that it's an infection. And in fact, clinically speaking, the phenomenon of what we call bacteriuria, which is basically just the presence of bacteria in the urine, doesn't require treatment, because clinically it's not considered an issue because the patient isn't showing the symptoms. Okay? And generally, the immune system, as well as the defense mechanisms of the urinary tract, including simple urine flow, will usually take care of that problem over time. But what about the long-term stented patients? Well, prolonged indwelling time results in bacterial biofilm formation. And of course, as I showed earlier, if that biofilm consists of a urease positive incrustation is a problem. So what happens in terms of biofilm formation? Well, you get individual bacteria attaching to the surface. These then start to multiply into what we call microcolonies. These microcolonies then begin to produce exopolysaccharide. So basically what you get is this extracellular polymeric substance, which is basically carbohydrates, so sugars, polysaccharides, and junk. Anything the bacteria doesn't want. Waste DNA, if you will, it just puts it all in there. And that is exactly what caused the problem. It's because that thick protective layer is basically what prevents antimicrobial agents from penetrating through and killing the underlying bacteria. So that's why bacterial biofilms are such a big problem and very difficult to treat. And then that process just repeats itself. Eventually, that device is removed, produce biofilm all over and obviously chronic infection in the patient. So, what has been done in the past to try and combat that issue? Well, here again is the antibiotic pretreatment of hydrogel, of the hydrogel that covers the ureteral stent. So the idea behind the hydrogel is mostly to make it more, to add lubrication to the surface of the stent, to make it easier to insert. Okay? And so what this particular group did is they pre-treated these stents with antibiotics in the hopes that the antibiotic will be absorbed into the hydrogel. They then tested it, and they actually showed that hydrogel coating and antibiotic pre-treatment actually resulted in greater bacterial numbers, which is quite interesting. I mean, this is likely due to the fact that the antibiotics were never really retained. Okay. But it was a good try. You know, it kind of makes sense. You know. Then the next obvious attempt was to give oral antibiotics. In this case, it was fluoroquinolones, because at the time that's what you did for urinary tract type infections. You gave fluoroquinolones. So in this case, patients were given either ciprofloxacin or ofloxacin over the indwelling period of the stent. And they found that in this case, the antibiotics were quite readily absorbed to the film covering the stent at concentrations that were higher than the minimal inhibitory concentration for E. coli. So that's a bonus. That's a good thing. And in fact, I mean, they did find no viral bacteria on the surface of the stents in any of the patients. There were no reports of UTIs. So that was promising. However, if you look at when the study was done, it's probably too small for you to see it back there, but that was 2001. I have to tell you that resistance to fluoroquinolones has skyrocketed since then. It is a big problem. In some areas, ciprofloxacin can no longer be used because of that reason. So using antibiotics always is a danger of increasing resistance. Now, obviously, there is a time and place that antibiotic treatment is required. Diamond-like carbon coatings was something else that was attempted. This was tested in 10 patients requiring long-term stenting because of malignancies. These patients also had a history of bacterial biofilms, associated encrustation, and symptomatic urinary tract infections, resulting in stent changes within 6 weeks. So every 6 weeks, they would have had a stent change. So over the indwelling time that these diamond-like carbon-coated stents were tested, which was mean time of 97 days, they didn't find any crystalline biofilms in any of the patients. And the frequency and severity of symptomatic UTIs was also decreased significantly in all patients. But if you look at the market, that stent is nowhere to be found. So for whatever reason, it's not being made. Now the reason I don't know. But this was a promising attempt at preventing encrustation and bacterial biofilm.
Is it because of the cost or manufacturing?
It probably is,
or
absolutely.
All of the above, or?
I would have thought. I mean, you know, that's why the majority of great ideas, you know, fail by the time they get to industries, because they don't want. We've had this conversation before. You know, if you come up with a new coating, a new material, whatever, you better do your homework and make sure it doesn't cost the companies more than what they currently pay, because they're just going to shelve it. You know, because they don't want to spend more money.
What's the typical time for a stent to be in? So that's like that's a long one, right?
Yeah.
Is that
So the majority of patients will have it in for, on average, about 1 to 2 weeks.
okay?
So, but the issue is most serious in the long term.
Yeah, I got that. But that might be also contributing, right? Because if there's fewer long-term,
Right.
then,
No, of course.
then the carbon-coated one, which would work there, isn't needed for short-term.
Sure, but you know, when you look at the market, there are actually specific stents that are made for malignancy. So patients with malignancies.
Oh.
So there is a market for it, but you know, I'm certain that sure that cost probably has something to do with it. So, oops, never talked about this. So this is something that came out first in 2009, but it's still being worked on. Is sort of stents that were coated with phosphorethylene glycol, but the way they attached it was by using what's called DOPA. And it's basically what marine muscle is used to attach to surfaces. It's kind of cool, actually. So they use that to attach the peg to the surface of the stent. And as you can see, both in vitro and in vivo, there was a decrease in the number of adherent bacteria. So, which again, was very promising. Again, this group is still working on this. So, it remains to be seen. To my knowledge, it hasn't been tested in humans yet. So, it'll be interesting to follow through some more to see how that pans out. So drug-eluting ureteral stents. Well, again, they kind of made their mark in vascular stenting. Drug-eluting stents did many years ago. They worked very well. So the idea was, excuse me, let's try them in the urinary tract. Of course, the idea is for the stent to elute an antibiotic to attack planktonic or free-swimming bacteria to kill them before they can attach to the surface. So I introduced you to the triclosan stent. So this is kind of when I started to enter the field, when the triclosan stent was out. So I started doing some of my work on this triclosan stent. And triclosan inhibits bacterial fatty acid synthesis. It's broad spectrum, both bacteriostatic and bactericidal. It's anti-inflammatory. And it's been used for more than 40 years. Toothpaste, soaps, surgical scrubs, children's toys, who knew? Right? So it's found everywhere, it's been used everywhere. So it was impregnated into a ureteral stent. It was called the Triumph stent. What was found in initial studies is that there were high concentrations at the device surface, concentrations that would be antimicrobial. So the first, this particular study is the one I was referring to earlier, actually. Just to go back to the aberrant coated stent, that was the radiant stent, by the way, and you can see that. Against E. coli, Klebsiella, Staph aureus, and Enterococcus faecalis, there was absolutely no difference between an uncoated and heterocoated stent. But the one that did work really well in all cases was the triclosan eluting stent. So the Polaris stent was basically the same type of stent, but without the triclosan floated. And you can see it was quite very effective against E. coli, Klebsiella, Staphylococcus aureus, and even Enterococcus faecalis, which are the major some of the major uropathogens. Okay, so this was then also repeated by another group, which again showed, and this graph shows percent reduction in bacterial adhesion. And again, you've got even Pseudomonas aeruginosa in this group, as well as Proteus mirabilis, and you can see again, 40 hours and at 96 hours, highly effective at killing the bugs, which was nice. Pseudomonas didn't work as well against, but that was basically mostly due to the fact that Pseudomonas is naturally resistant to certain concentrations of triclosan, so it wasn't necessarily surprising. And then on the right here are images, scanning electron micrographs, basically showing uncoated and coated samples over time. And certainly the coated ones have less bacterial numbers entering off the surface. So the next logical step is, well, let's try this in vivo. Now, at the time, the rabbit model was very popular. It's not popular anymore, mostly because rabbit urine is very thick and very viscous. But at the time, it was the acceptable model. So the idea was to take basically the J ends of the stents and and basically suture them into the bladder of animals and induce an infection and see after a certain amount of time how many bacteria are attached. And incidentally, these were rabbits were infected with Proteus mirabilis. Why is that significant? Well, Proteus is urease-positive bacteria. So here, not only could you look at decreased infection, but you could also look at decrease in the device surface. So the end result, triclosan eluting stents significantly decreased bacteria from urine in the majority of the animals within 7 days. You can certainly see that in the graph here. So there's day 1, day 3, day 7. You can certainly see that the bacteria are less. There's significantly less bacteria on the triclosan eluting stent than the other control stents. Now, when you looked at them up close, 7 out of 13 actually had no bacterial growth at all. So, which was very promising because, again, if you think about it, these animals were challenged with probably 10 to the 8 bacteria to begin with, and to have nothing left on the surface after 7 days is quite remarkable. So we got all excited and happy. Finally we're getting somewhere. And so now you move into humans. And it's a whole different ballgame. Because what we found in humans was that, unfortunately, there was no significant difference for culture and incrustation results. Okay. And again, you can see here. Now, admittedly, it was only done in 10 patients, but it's a small trial like this can even make or break a promising product. So anyway, you can clearly see that, again, no differences in terms of cultural positives or incrustation results. However, subjects in the triclosan group did report significant reductions in lower flange pain and abdominal pain scores during activity, as well as urethral pain during urination. So that is the only time that I'll be talking about the whole discomfort aspect of ureteral stents and that whole complication, because this particular stent clearly showed a decrease in terms of discomfort scores. So that was a plus. Now, why is that? Well, that's because, as I mentioned before, triclosan also has an anti-inflammatory effect. And in this particular study here, what we did was we wanted to try and understand the pro-inflammatory cytokines secreted in response by both bladder and kidney epithelial cells in response to stent irritation. So we developed an in vitro model in which we tested the triclosan eluting stent or the control stent, but added triclosan to the actual medium, and we quite clearly showed reductions in significant reductions in pro-inflammatory cytokines when you both bladder and kidney epithelial cells by triclosan. So the first study in humans I showed you there, that was short-term stent patients. So, but what about long-term stent patients? And again, in this case, there was no clinical benefit in terms of urine and stent culture for both the controls. Interestingly, this is definitely a plus. There was decreased antibiotic usage and significantly fewer symptomatic infections, which definitely is a plus, because basically what we had more of was patients perhaps with bacteriuria, meaning no symptoms. But then again, the question is why did the symptoms go away? Is it the anti-inflammatory effect of triclosan, or did it actually do something with the bug to reduce them to certain numbers? Well, we never know, but the conclusion is that the triclosan eluting stent alone is not sufficient to reduce device-associated infection. And it was because of that, as well as the fact that the FDA said, well, you know what, we're very concerned about the increase in triclosan resistance, despite the fact that it had been used for 40 years. It's because of those two reasons that this stent never, well, actually did make it to the market, but it was taken off the market and is no longer being made. In terms of making progress, I still maintain that that was the closest we've ever gotten to stent that actually went up against two of the big stent-associated complications, which was obviously infection and patient discomfort. And potentially incrustation, because, you know, when you decrease infection, then it's true like incrustation
Is a triclosan coated stent treated as a drug or combination product, or is that a device?
I believe it's a combination product.
Combination product.
Yeah. So then the question that arose was why are surface coatings not effective? And that's where the role of the conditioning film. Okay, and what is this urinary conditioning film? Well, as I mentioned at the beginning, urine is full of all kinds of components. So as soon as you introduce any device into the urinary tract, what starts to happen is that these components start to attach to the surface. Well, what does that do? Well, it covers your novel antimicrobial coating or drug-eluting technology and basically renders it ineffective. The other hypothesis that was out there is that while doing that, it probably provided bacteria a pedestal for adhesion and biofilm formation, because bacteria produce adhesins that recognize and bind quite readily to a lot of these components that are found in urine. A lot of them are proteins. Yes?
What about the surface topography of the stents, you know, the smoothness, and how does that come to play? Because there are now startup companies that they're taking advantage of the nanotechnology to make rough surfaces to prevent bacterial adhesion.
Yes, and so the stents are supposed to obviously be smooth, but I mean, when you take an SEM image of a stent that hasn't been inserted in a patient, you can quite readily see that there is certain unevenness in coatings as well as obviously the surface material. So that does play a role. However, even if you look at some of the papers that have been published on some of these novel patterns on biomaterials, what it does do is it does decrease overall biofilm formation. In terms of bacterial adhesion, I'm not convinced that it necessarily decreases that, because the idea behind it is that an uneven surface basically just breaks up biofilms from conjoining, but it doesn't prevent necessarily the microcolony formation. Okay, so that's what I would say about that. I'm not suggesting it wouldn't necessarily work, but I think in the long run, I'm not convinced it would make a significant difference. So, so the next step was to try and understand this conditioning film. You know, what is in this conditioning film? Because we didn't understand anything. We accepted that it happens, but what is it? And so we started looking into that, and what we did was we started taking stents that were removed from patients. We started analyzing what was in the conditioning film. Now, we compared it. We compared conditioning film on the same stent between patients, but we also compared different types of stents because we wanted to understand, is the conditioning film similar between patients on the same stent? And then is it similar on stents made of two different materials? The overall conclusion from this study was that there is no significant difference in either case. The major adhering components were basically all the same. And, you know, they're all listed here, and it's quite interesting. There, you know, cytokeratin, serum albumin, hemoglobin, uromodulin, which is the most abundant protein in urine, fibrinogen, and so on and so forth. So a lot of these were actually proteins associated with either blood or inflammatory response. So why were they in the blood? Well, it could be because of the surgical procedure, you know, or of course they might be naturally in there as well, just feel like they are. But the point is, there's no difference. Interesting enough, in terms of aiding incrustation, because again, we don't quite understand how that necessarily works. You know, how do these crystals attach to the surface? We did identify some calcium binding proteins which may facilitate that incrustation. Again, at this point, it's still just a hypothesis. So if a way that we could maybe prove that, that would be outstanding. I think that would be a pretty good step. So the question is there a role for conditioning film and bacterial adhesion? I mean, that was a big question. And I got to tell you there isn't. Because again, we tested E. coli and Staph aureus adhesion to Polaris control or Polaris removed patients, and you can clearly see that there's absolutely no significant difference. Okay, sorry about that. There's absolutely no significant difference whether it was conditioned or not. So there we go. The concluding thought for that then. Conditioning film may prevent drug elution and inhibit novel coatings, as I discussed before, but there is no role for increasing bacterial adhesion and colonization, or as originally thought. Now, again, its role in crustacean remains to be determined. Now, there is no arguing, though, that it does still facilitate bacterial interaction with the surface. So where do we go from here? That's a good question, because we are in fact disoriented, bewildered, perplexed.
Like how we operated.
Yes, exactly. I mean, that's right there is what research is all about. So what has been tried since? Well, you know, there's been a move towards biodegradable materials. Okay, here's an example of a modified polylactide surface with polyquaternary ammoniums, which is all fancy terminology to me, but I'm sure it makes sense to you guys. Okay, and the idea was for it has broad spectrum bactericidal activity, and the idea was for it to come out as the surface degrades, and sure enough, you can certainly see that the bacterial numbers are significantly decreased, as is bacterial biofilm formation. So again, very promising. At this stage, it's only in the in vitro stages, so it'll be interesting to see what happens thereafter. Here's another example. This group coated silicone-based ureteral stents with tocopherol acetate, which is a biodegradable polymer, and they impregnated that with ofloxacin or nudazole to antimicrobials. And again, in vitros, you can see that this zone of inhibition quite clearly indicates that, you know, as the material degrades, the drug certainly gets rebelled, and so it seems to work. Again, it's only in the sort of in vitro type stages. So the question is there actually room, though, for biodegradable ureteral stents? I mean, this is really what we're talking about. Is such a thing possible? And I'm here to tell you that yes, it is, because we've been working on it. Okay, and so we've been working on a biodegradable stent for a few years now. It's gone through different generations. The original one took about 10 weeks to degrade. The latest takes about 28 days to degrade, which is pretty much the ballpark you want to be in. Why is a biodegradable stent more advantageous? Well, a couple of reasons. One, degrades by itself. So that means the patient doesn't have to come back in and have that stent removed, meaning there's no procedure required for this, which does cause some sort of discomfort to the patient. Clinicians might not like it very much because they can't bill for an additional visit, but as far as in terms of the patient, it makes a lot more sense. The other thing too is a biodegradable material changes physical characteristics as it degrades. So there is the possibility, this hasn't been tested yet, but there is a possibility that a constantly changing surface may, at least to some degree, decrease the chances of bugs being able to interact with it. Because as smart as bacteria are, I don't think they can change their surface characteristics quite as quickly as something that degrades relatively quickly. So these studies here that you see here were done in pig models. We have recently completed a small clinical trial. I'm happy to say that the stent performed as expected without any sort of significant complications in the number of the patients tested, and so we're looking to do a larger clinical trial on this. Now, the advantage here as well is to come back to the whole drug delivery idea, because a biodegradable stent like this could quite easily be loaded with some sort of drug or polymer or what have you that could be delivered as it degrades to prevent potential infections or anything like that. So that's certainly an advantage of that. Another approach that we've been quite active in is the whole idea of moving away from antibiotic use. So we've been collaborating quite closely with polymer chemists and antimicrobial peptide expert, Dr. Bob Hancock at UBC, on using antimicrobial peptides and linking them to surfaces. And so the idea here is that, as I mentioned before, conditioning film will cover up anything on the surface. But the idea here with the peptides is to move the active ingredient away from the surface, so it acts away from the surface. So even if conditioning film components start to deposit on the surface, it doesn't matter because the active ingredient still works. And so what we have is we have a substrate, we have a polymer linker to which we attach the peptide. Oh, sorry, and just to come back. So the advantage of using antimicrobial peptides, of course, is that they're nature's antibiotics. They're part of innate immunity. There is, I don't want to say virtually no resistance. I mean, there's a little bit of resistance, but certainly not as much resistance as there is to antibiotics. And the type of resistance to antimicrobial peptides can be acquired as easily as antibiotic resistance. And they're relatively broad spectrum, okay, and some of them are also immunomodulatory, meaning that they will stimulate certain areas of the immune system and could be quite beneficial as well. So
can I there is a concern that the antimicrobial peptides would actually mess up with the innate peptides, and so the FDA at some point might step in just like it happened with the triclosan. Yes.
Oh, they might.
People have been speculating that
It could be a problem.
it could be a problem because
Yeah.
each also uses them. So if there is resistance to them, that would actually be more problematic.
Well, but you could argue that nature also uses antibiotics because nature, antibiotics kind of came from nature, at least some of them, right? So it's kind of, I know. I mean, the FDA unfortunately always puts huge hurdles in your way. But I'm not suggesting that down the line, I mean, it's like anything, right? When we start using antibiotics, there was no resistance until we started using them once. So it's quite possible that now that, if we start using antimicrobial peptides a lot as well, that something might come up. This, certainly can happen. So, but in the now, it worked quite well. Again, you can see quite readily that in vitro, the peptides certainly, surfaces that had peptides linked to them had significantly less bacteria. And what was even more astounding, which I wasn't actually necessarily expecting, is that it effectively eliminated bacteria in solution. So these peptides weren't designed to come off. And so they didn't come off. But they still actively killed whatever was in solution. The theory is that it'll kill whatever comes into close proximity to it, so, which was quite amazing. Again, it shows significant anti-biofilm activity against Pseudomonas aeruginosa, and the in vivo testing also showed that the percent decrease in colony-forming units on implants was significantly less than all the animals tested. And so, in this particular case, by the way, this was titanium implants, and so we were working on sort of implant infections and things like that. So the next step was to actually, obviously, try this on urinary biomaterials. But there was one limitation. And that limitation was there was no proper in vivo model. You know, there's all kinds of innovative catheter materials, novel antimicrobial coatings, and so on and so forth that come out. But they've mostly been studied in vitro, and certainly some of the promising results I just showed you were done in vitro. And just like a lot of areas, there's a general lack of reliable, cost-effective, and high-throughput preclinical animal models. So in terms of a urinary tract infection model, the limitations of the models that were out there were, there was an issue with reproducibility. So the way that they would introduce a biomaterial, and I mean, we tried this as we developed, is you would just basically take a small catheter piece, essentially, insert it through the urethra, just like you would do in a human being, and then leave it in the bladder. But the problem was that that piece, that catheter piece, in the majority of animals, was never inserted all the way. So part of it stuck in the urethra, so when the animal went to urinate, it just peed it back out. So you had a loss of material in more than 50% of the animal. So that's not very cost effective. Of course, it's not very good because you have to use more animals to study these things. And of course, well, it might not be as obvious to you, and it wasn't to me initially, but it's restricted to female animals because male animals have a spiral urethra. Well, not all animals, but rodents do. Don't ask me why. I don't know. The other issue too was there were other models out there which were highly invasive. Just like the rabbit model, what you had to do was basically open up the chest cavity. Well, not the chest, but lower down. Find the bladder, make an incision in the bladder, put the piece in, suture it in place, suture up the bladder, suture up the animal, have the animal recover, then infect. I mean, this took, not only did this take a long time, but just look at the surgical procedures you've just done. The chances of secondary infection, sepsis to the animal is significantly high. So it's not a very good model. So our objective was to establish a simplified, reproducible, minimally invasive model. And initially we developed it for catheters associated with urinary tract infections and encrustation studies. Our model is ultrasound guided, which means you can see pretty much every step. And it's applicable to both genders because we don't go through the urethra. Essentially what we do is we use a percutaneous approach. So basically what you do is we use an angiocatheter, and we've all seen these, you know, when you have an IV placed, right? So we cut the tip of it off. It's about half a centimeter, I believe. Then the next part is through ultrasound guidance. We then locate the bladder, and you insert the needle into the bladder. And you can see it in pictures quite nicely here. So, needles in the bladder, and you can certainly see the needle in the bladder. The ultrasound image, that's the outside of the bladder, here's the needle. You then advance the plastic piece, which then basically launches your test material into the bladder, and you can see quite nicely here. You can then remove the needle and inject your bacterial of choice, which is quite convenient. Then you remove the plastic and you leave everything intact. The piece in the bladder. Nice thing is that the bladder wall is actually quite resilient. It just seals itself off, so there's no leakage or anything like that, so which is good. So in our case, technical success rate has been 100%. We haven't lost any catheter pieces, which is very promising. And so we just recently used this to test antimicrobial peptides linked with the polymer brushes to catheter materials. And as you can see up top, you've got untreated control animals and treated animals. Okay, this is day one. Basically just showing you that all the animals had infection. Now, why do the bladders light up? Well, they light up because the Pseudomonas strain that we use to infect them actually expresses lux, so you can detect it quite nicely. When you look at this in the graph form, you can see that the total flux, again, between uncoated controlled mice and coated mice is basically the same. So we all started at the same baseline. Well, when you look at 7 days post, you can certainly see untreated control mice got significantly worse because it gets bluer, there's more bugs. Whereas the treated mice, you can see that the majority of them actually cleared the infection. And certainly, again, when you look at the total flux numbers, you see significant decrease in the animals that received the coated catheter pieces. But like a true microbiologist, looking at fluorescence or whatever tags is always nice and wonderful, but I want pure, hard CFU counts. That's what I want to see. And sure enough, as you can see with the colony forming units at day 7, you could still see that there was a significant decrease in numbers found on catheter pieces coated catheters versus not. So, to conclude, we did have an infection at day 1, which is good. Everything was good, baseline. And the polymer crushed tethered peptides did significantly decrease bacterial load. That was point 2. It was also, on the surface, so on the catheter piece itself, as well as in the urine. So again, planktonic bugs were all killed, which is astounding. Still boggles my mind. This was all done at day 7. So the results look very promising. However, as I've learned, exposure to urine, human urine will pave the way to success or failure. So we'll see what happens. And with that, I would like to thank all my colleagues, because of course I can't do all this by myself. Dr. Ben Shu is my clinical colleague. He's an endourologist that I work very closely with. We run the STEM Center clinical and basic science research program, so the translational research program. Jay Kizakidatu is the polymer chemist that has a collectively. Bob Hancock, of course, antimicrobial peptide expert. Claudia was the postdoctoral fellow who did a lot of the mouse model development. Igor Moskalev, how do you say it? It's not Moskalev, it's Moskalev. He taught me, well, no, he didn't teach me, but he is brilliant animal techniques. He also assisted with the ultrasound guidance. Dr. Kai is postdoc in Jay's lab, and then Joy Lowe is my master's student who's been doing a lot of work as well. Up top's Vancouver. Down below some pictures of orcas that I took recently on a whale watching trip. And yes, I did take the picture of that bear, and the one thing that I did learn is do not get that close to it. because about 10 seconds after that, it stood up and I ran. So with that, I'll take any questions.
Wonderful.